Well, welcome back. We've been looking at genetic information recently and how that genetic information is stored on DNA. But have you ever wondered how that genetic information gets turned into the actual traits that you and I can observe every day, such as the color of our hair or our eye color? Well, that's what we're going to begin to focus on in these next few segments. And right now, I want to introduce to you a very important molecule known as RNA. You see, every gene that's on the DNA molecule is going to be converted into a much smaller molecule, a nucleic acid, that's going to carry the information of that gene into the actual production of the trait that you and I see every day. So let's discuss this topic of transcription. And as you can see, transcription converts a gene into a single-stranded RNA molecule. All right, now there's a larger backdrop to this whole story here, and sometimes referred to as the central dogma of molecular biology. And that simply states that information flows in one direction from DNA that you see here to RNA to protein. And so it's really the proteins that are going to produce the traits that we can see expressed in organisms. They do the day-to-day in and out operations of the cell, structural components, and so forth. You'll notice, just by way of review, that there's a loop here within the nucleus of the cell. And just recall that we recently looked how DNA makes copies of itself, known as replication. Well, that process in some ways is going to be reflected in how RNA is made, which we'll look at next. So we have these three processes: replication of DNA within the nucleus, transcription, in other words, converting some of this information onto the RNA molecule, and then translation, the conversion in the cytoplasm of that genetic information on an RNA molecule into the actual proteins that either perform that trait or produce that trait. So that's where you see them all come together. And so RNA is really the link that's between the DNA and protein. Now, RNA differs from DNA in three major ways. First, RNA has a sugar known as ribose instead of deoxyribose, hence the change of the name. Another very important change is that where we saw thymine as a nucleic acid in DNA, that's now going to be replaced by a different nucleic acid known as uracil. And thirdly, RNA is not a double-stranded helix such that we saw in DNA, but it's a single-stranded structure. And so those nucleotides are going to be exposed on the RNA molecule as they're going to be quickly accessed. All right, and here is a diagram form of that nucleic acid uracil. You can see that this is a pyrimidine. And this process of transcription is catalyzed by an enzyme known as RNA polymerase. You may recall that term polymerase when we were looking at DNA replication. This is a similar kind of enzyme, but it operates to build an RNA molecule. So RNA polymerase and other proteins form a transcription complex, and that complex recognizes the start of a gene and unwinds a segment of DNA so this information can then be copied. There are free nucleotides that are going to float around in the nucleus, where these will be used in the production of RNA. Now, nucleotides pair with one strand of the DNA, and then the RNA polymerase bonds these nucleotides together. Recall that the backbone of DNA is going to consist of covalent bonds. That's also true in the RNA molecule as well. And so the DNA helix is going to wind again as the gene is copied or transcribed. As you see here, RNA polymerase is going to move along the DNA molecule in much the same way that DNA polymerase did. In this case, however, only producing a single strand molecule. And then the RNA strand is going to detach from the DNA once the gene has been copied or transcribed, as you see here.
What you are about to see is DNA's most extraordinary secret: how a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
And transcription makes three types of RNA. The first is messenger RNA, which carries the message that's later going to be used to produce a protein. There is ribosomal RNA, which forms part of the ribosomes where proteins are made out in the cytoplasm. The ribosomes themselves will be manufactured within the nucleus itself in the nucleolus. And then finally, transfer RNA. These are molecules that will actually shepherd the amino acids from the cytoplasm to the ribosome so that proteins can be made. So transcription process is similar to replication in that both involve complex enzymes and complementary base pairing. Remember those base pairing rules? Well, they're also going to apply here with the one noted change previously of thymine being replaced by uracil. The two processes have different end results, however. Remember that replication is going to produce DNA, transcription is going to produce RNA. Okay? Replication copies all the DNA, transcription copies the gene. So it's a much more limited amount of information. And replication makes one copy, whereas transcription can make many, many copies so that many proteins can be made simultaneously. And here we see a picture of this using the benefit of an electron microscope. How you can see one gene now is going to be used to transcribe many DNA, is going to be used to produce many RNA strands from that one gene. Well, thanks for watching. This was transcription, the production of RNA. We'll see you soon. Bye-bye.